Hello, and welcome to the first webinar for the Chemical Sciences Roundtable at the National Academy of Sciences for 2025. In what I can only assume is cosmic timing or perhaps brilliant planning, we find ourselves gathering on World Quantum Day. Yes, today is 414, and the first three digits of Planck's constants are 4.14. For those who haven't had their coffee yet, that's the fundamental constant that relates the energy of a photon to its frequency. And there's some confusion about the units. As was pointed out to me earlier today, it can be expressed as 6.63 times 10 to the minus 34 joules per hertz, or in electron volts, it is 4.14 times 10 to the minus 15th electron volts per hertz. So let's start the slides. Now, while I can't promise we'll be diving into quantum tunneling or explaining why Schrodinger's cat is simultaneously attending and not attending this webinar, I can guarantee that with our great lineup of researchers, we're in for fantastic discussions today. So whether today's cosmic coincidence was intentional or just the universe having a chuckle, I think we can all agree that when it comes to today's panel, we should definitely expect the unexpected. Let me just point out the folks on our side that helped make this pull this webinar together. I'm Mike Janicki. I'm the director of the Chemical Sciences Roundtable, and via Zoom, emails, and IT wizardry, Kay, Eric, and Darlene deserve most of the credit for setting up this event. I'd also like to take this opportunity to introduce the Chemical Sciences Roundtable, chaired by Marty Burke and Nikki Pohl. I don't want to take time away from our panelists by introducing everyone on the slide, but I wanted to point out that the roundtable is supported by this amazing team of volunteers, and we very much appreciate all the time that they give to this activity. And I highlight the three members in particular today, Susanna, Mike, and Jack, who helped with the planning of this webinar. Now, this will be the first of three webinars for our topic on chemistry in 2050 that will conclude with a workshop hosted in Washington, D.C. next winter. Please sign up at our website to stay in touch with our activities. In the fall, we'll be having two webinars. One will be looking at education and workforce development, and the second one will be looking at, what future resources do we expect for in chemistry and chemical sciences, chemical engineering in the future. Ground rules for today. The audience, your cameras and mics will remain off. This webcast is being recorded, and we are using Slido to present questions to the panel. With the QR code, you can find a link to the Slido page. The web page address is below, and you'll also be able to access it during the webinar. So it is essential to the National Academy's mission of providing evidence-based evidence that participants in any of our meetings or events avoid political or partisan statements or commentary and maintain a culture of mutual respect. The statements and presentations during our meetings or events are solely those of the individual participants and do not necessarily represent the views of other participants or the National Academy of Sciences. One last item before we get started. The National Academies are committed to the principles of integrity, civility, and respect in all of our activities. We look to you to be a partner in this commitment by helping us to maintain a professional and cordial environment. All forms of discrimination, harassment, and bullying are prohibited in any National Academies activity. This policy applies to all participants in all settings, including this webinar. Just briefly for today's agenda, we'll have about an hour to hear from the background and perspectives from four great panelists. Then for about 20 minutes, Edward Ashton will help moderate a discussion, and then we'll pivot to a Q&A from the audience. So before I pass over the mic to Edward Ashton is a cancer researcher and expert in brain imaging, and was also the author of Mickey 7 that was turned into the recent movie Mickey 17. And so I will pass the mic to Edward, and we'll begin our panel discussion.
Thank you, Michael, and thank you all for taking the time to speak with us today. We really do appreciate it. Michael's opening remarks really emphasized for me something that I was aware of previously, which is that I am definitely the least knowledgeable person in this discussion. That's okay. I'm hopeful that I can at least provide some comic relief. But along those lines, let me just introduce our panelists for today. First, Dr. Jennifer Bergner. Jenny Bergner is an assistant professor in the Department of Chemistry at UC Berkeley, with joint affiliations in the Department of Astronomy and Lawrence Berkeley National Lab. She's an expert in astrochemistry with a focus on understanding how the composition and potential habitability of nascent planets are influenced by the chemistry at play during their formation. In recent years, she has been recognized with the Annie Jump Cannon Award from the American Astronomical Society, the Chevron Chair in Chemistry at UC Berkeley, and a Scilog Fellowship from RCSA. She completed her PhD at Harvard, followed by a NASA SIG and postdoctoral fellowship at the University of Chicago. Jason Dworkin is the senior scientist for astrobiology at NASA Goddard Space Flight Center and the project scientist for NASA's OSIRIS-REx mission. He's the founder and director of the Astrobiology Analytical Laboratory at GSFC, which employs modern analytical methods to examine authentic samples of the early solar system, as well as laboratory models of ancient environments. He has 40 years of experience in assessing the organic species available for the origin and early evolution of life, with a focus on understanding the extraterrestrial input and origin of organic molecules relevant for life. This objective has been to study increasing documented and constrained systems, from plausibly early Earth chemistry, chemistry of astrophysically relevant laboratory ices, organic and chiral analysis of meteorites, to analysis of sample return materials, how to protect the material from contamination. He has received numerous awards for his work with OSIRIS-REx and for science leadership at GSFC. He received an AB from Occidental College and PhD from the University of California in San Diego, both in biochemistry. Sara Seager is an astrophysicist and a professor of physics, professor of aeronautical and astronautical engineering, and class of 1941 chair professor of planetary science at the Massachusetts Institute of Technology. She's known for her pioneering research on exoplanets and their atmospheres. Her research has introduced many foundational ideas to the field of exoplanets, planets orbiting stars other than the sun. She's now at the forefront of the search for the first Earth-like exoplanets and signs of life on them. In addition, she's pursuing exploration of Venus as a habitable world through laboratory experiments of biochemicals in concentrated sulfuric acid. Professor Seager earned her Bachelor of Science in Math and Physics from the University of Toronto and her PhD in Astronomy from Harvard University. For her research, Professor Seager was awarded the MacArthur Genius Grant, the Kavli Prize in Astrophysics, one of Canada's highest civilian honors, appointment as an Officer of the Order of Canada, membership in the National Academy of Sciences, and has asteroid 9729 named in her honor. She's the author of The Smallest Lights in the Universe, a memoir. Jennifer L. Talley is a program officer of space bioscience at the Air Force Office of Scientific Research in Arlington, Virginia. As program officer, she discovers, shapes, and supports basic research that will benefit the Air Force, Space Force, and more broadly, the Department of Defense. Dr. Talley became an Air Force civilian researcher in 2011. She was a senior research biological scientist at the Integrated Seekers and Processing Branch, Guidance Division in the AFRL Munitions Directorate at Eglin Air Force Base in Florida. Some of her major awards and decorations include 2023 Research Excellence Award, 2022 Demonstration Award, 2020 Notable Achievement Award, 2019 Special Active Service Award, 2018 AFOSR Star Team Member, 2017 Outstanding Mentor Award, 2017 Windows on the World AFOSR Award. In 2019, she was listed as a co-inventor on U.S. patent number 11,703,880, for biohybrid odor localizing autonomous air vehicle. Prior to her current position, Dr. Talley earned a master's in strategic studies at the Air War College and Residence, Grand Strategy Seminar at the Air University, Maxwell Air Force Base, Alabama. Dr. Talley received a PhD in biology from Case Western Reserve University in 2010 and a BS in mathematics from the University of Arizona in 2003. And with that, I'll turn things over to Dr. Bergner to kick things off.
Thanks for the introductions, Ed. So my name is Jenny Bergner. I'm an assistant professor in chemistry at UC Berkeley. And as a little background of how I got into astrochemistry, all of my degrees are in chemistry, and I discovered astrochemistry towards the end of my undergrad education, going to an internal seminar by an astrochemist. And it kind of blew my mind because the talk that I saw was introducing all of these molecules that after 4 years of chemistry training, I thought shouldn't really be able to exist. So I was initially drawn to the sort of exotic weirdness of astrochemistry. But then as I got more and more interested in it, I became really much more interested in not just understanding the weird chemistry for weird chemistry's sake, but the implications that it has for really profound questions like how habitable conditions emerge on planets around other stars. So the research that I do today is really focused on chemistry in the context of star and planet formation and what we can learn about how the compositions and properties of new solar systems are shaped by the chemical environment that they form in. So I will share some slides now. Okay, so as a little bit of context, the formation of new solar systems happens within these disks around young stars. So forming planets is sort of a byproduct of forming new stars. And the material within these circumstellar disks is made up of dusty things, sort of similar to silicates or refractory carbon. There's also volatiles, so molecules like water and carbon-bearing species that we're especially interested in from the context of habitability. So we know that or we expect that life should require water, as well as access to elements like carbon, nitrogen, oxygen, and so forth. And so we can study these regions of space where planet formation is ongoing to try to understand how the building blocks for life and habitable conditions are incorporated into planets. So of our panelists, I think my research area is sort of at the beginning of this journey of going from the very building blocks of planets all the way through to sort of fully formed exoplanets and life and so forth, as we'll hear about from some of the other panelists. So I see the motivation as sort of twofold, actually. On the one hand, we're very interested in the chemistry at play in these astrophysical environments and how this affects the compositions and potential habitability of newly formed solar systems. But also as a chemist, I'm quite interested in the processes that are taking place in these extreme environments. And all of the rules of chemistry are the same, but the outcomes can be really different because the physical conditions are so different. And so I'm interested in what we can learn about fundamental chemistry by studying chemistry in these extreme environments. So as a little bit of context, chemistry in space happens under really extreme conditions. The temperatures can be as low as 10 Kelvin, which for those of you less familiar with the Kelvin scale, that translates to negative 450 degrees Fahrenheit. And sort of at best, we're around 13 orders of magnitude lower pressures than the atmospheric pressure we have here on Earth. So these conditions, as I said, are quite extreme, and that can really dramatically influence the types of reactions and the types of processes that will take place. And in turn, sort of the composition of the materials that are available for coalescing into planets. So one way that we can try to understand the chemistry going on in these extreme environments is through lab studies. So we can try to produce environments mimicking the extremely low temperatures and pressures of space in the lab. This is an image of the lab that we're building at UC Berkeley in my group. So the center sort of sphere that you see, this is a vacuum chamber. And within this vacuum chamber, we have really powerful pumps that evacuate it to sort of space-like pressures. So 13 orders of magnitude lower pressures than the air around us. And then suspended at the center of this vacuum chamber is a cold sample. So that's shown here on the left. And we can actually cool this down to temperatures as low as 8 or 10 Kelvin. So we're really mimicking the extremely frigid conditions of interstellar space. And then with this very controlled physical environment, we can study how molecules behave. So in these kinds of conditions, a lot of the species that we're interested in, like water, carbon-bearing volatiles, nitrogen-bearing volatiles, things that would normally be gases in Earth's atmosphere are actually, they exist in a frozen state, which we call ices. And so you can actually see a little smudge on the window of this cold sample holder that is an analog to the icy material that we expect to find in interstellar regions. So we can produce this analog interstellar material and again study how it behaves, things like how molecules react, as well as their microphysics, so how they move around, how they sort of partition between the gas and ice phase, and so forth. So the big thrust of the lab studies is to understand how molecules behave chemically and microphysically in these extreme environments. The other aspect that we can sort of bring in to try to understand this chemistry associated with planet formation is using really powerful telescopes. So for any chemists in the audience that haven't encountered telescopes before, we're basically doing spectroscopy, but on regions of space that are really far away from us. So hopefully everyone has heard of the James Webb Space Telescope. This is our new infrared telescope that launched just a few years ago. It's providing us beautiful images of these planet-forming regions. So in this case, we're actually looking at a circumstellar disk with an edge-on configuration. And so the disk is so dense that it's blocking out all of the light. So you see the sort of dark lane in the middle, but then we see light in the infrared being sort of scattered out along the flared surfaces of the disk and its sort of natal envelope. So these images are really gorgeous. We can also complement this with information from ground-based telescopes. So the ALMA array down in Chile is another workhorse for the field. And this allows us to sample the composition of gaseous species local to where planets are forming. And one of the really amazing things that we're seeing with ALMA is the amount of structure that's present in the distribution of different molecules. So this is an image of the molecule HCN towards a protoplanetary disk. So you can literally visualize how this disk might someday turn into a solar system if it was sort of, this material is coalesced and condensed into planets. But the really fascinating thing is that we see these sort of rings and gaps in the distribution of different molecules, and that's telling us a lot about the chemistry going on. So we love images. They're really easy to relate to, but as chemists, what we're even more excited about is the spectral information we get. So this is the spectrum of a protoplanetary disk with an edge-on viewing geometry taken by the James Webb Space Telescope. And you can see these absorption features from molecules like ammonia, CO2, and CO. And actually, James Webb is the first time that we've been able to make these measurements. We never had a telescope that was sensitive enough to do these measurements, which require really, really high sensitivity towards the protoplanetary disks where planets are forming. And so in the last couple of years, we've actually been able for the first time to put constraints on the compositions of the volatile icy material that is directly available for planets that are forming. So with this kind of telescope data, what we're interested in is constraining the composition of the building blocks of planets, and ultimately in understanding sort of based on the compositions of the building blocks, we can make predictions for the properties of other solar systems. So what are they like? Are they similar to our own solar system, or are they very different? So a couple of specific research highlights. I've done a lot of experimental work looking at the emergence of chemical complexity through interstellar chemistry. So we know that there's actually a wealth of chemical complexity that can form in interstellar environments. And this is interesting because this chemical complexity could potentially seed newly formed planets with building blocks for prebiotic chemistry. So understanding the mechanisms of forming these larger organic molecules, even in extreme interstellar conditions, is quite interesting. And so I've done some lab work putting constraints on new pathways to actually generate this chemical complexity. Another fun example is an application of sort of the fundamental microphysics of molecules and how they might affect the macroscopic behavior of planetesimals. So we had a study where we showed that the sort of peculiar non-gravitational accelerations of the first interstellar object, Oumuamua, can actually be explained based on the microphysics of water ice, and specifically the amorphous kind of water ice that we expect to be present in interstellar environments. So we can actually explain the non-gravitational acceleration through water ice microphysics without invoking any sort of exotic physics or chemistry. And then another sort of ongoing thrust is using James Webb to look at the composition of volatiles in protoplanetary disks and understanding, for instance, how much water, carbon, and oxygen are available to forming planets. So again, this is possible for the first time thanks to James Webb, and I'm leading a program doing a sort of demographic survey of the ice compositions in a handful of protoplanetary disks. So just a few words towards looking ahead. I think, especially for a chemistry audience, I think it's important to emphasize that astronomers really need chemists, and this need is only going to grow as the field develops. So we have these fantastic telescope facilities that we spend hundreds of millions or billions of dollars on. But if we want to actually interpret the data from those facilities, we need fundamental parameters like reaction kinetics, spectroscopy. We need to understand the microphysics in these extreme environments. And so I just want to emphasize that as our telescope facilities get better and better, we're only going to need better and better constraints on these fundamental parameters in order to actually make use of the data. So what I've shown here in an image is a spectrum towards a young planet-forming region taken by James Webb. And this spectrum allowed the discovery of the CH3 plus molecule for the first time, which is a driver of interstellar chemistry that was hypothesized to be present for a long time. But it was only with dedicated lab work and theory that this identification was able to be made. So you can see the really beautiful prediction on the bottom and how it matches to the observed spectrum. So again, we really need good constraints on fundamental chemical and physical parameters in order to have any meaningful interpretation of the data that we're getting from the telescopes. Another sort of area that I'm quite excited about is having observing capabilities in the far IR or terahertz regime. This is a region of the spectrum where we currently have no observing capabilities. So this figure shows James Webb at the shorter wavelengths, ALMA at the longer wavelengths. We saw images from both of these facilities a few slides ago, but we're missing a big part of the spectrum right now in the far infrared, and they're really key diagnostics of the volatile composition of star and planet forming regions that we basically can't access right now. And so one of the things that I'm excited about going forward is the possibility of filling this in with a space-based far IR mission. And there's a mission concept under study right now called PRIMA that would do that. So that's all that I had prepared. I'm happy to pass off to the next panelist.
Thank you, Dr. Bergner. Real quick question. Very disappointing. Oumuamua, not a spaceship, huh?
It doesn't have to be, no. There are plausible sort of natural explanations for its behavior.
That's fair. There goes my next book. Thanks. Great. Okay. On that sad note, let's move on to Dr. Dworkin. Take it away.
Hello. So that's a great place for me to start. And I just inserted this slide to get us going. If I can figure out how to do this. There we are. So we've just heard about what you can do by looking at the astrophysical environment and looking for origins questions. You can study the cycle from the diffuse medium, dense clouds, star formation, stellar death, and mass ejection as stars and planets are formed and die. You can also do simulations on either chemical or mathematical on primitive bodies to understand them. But I'm interested in what you can get in the laboratory, and by doing analyzing the witnesses of these fragments, the leftover pieces of the formation of the early solar system. So I've been interested in questions on the origin of life since about the second or third grade. And these are some of the main questions on how life maybe got going. The origin of homochirality, the left versus right-handed molecules, the sources of these compounds, the nature of the first information molecule, whatever preceded the DNA, RNA that we see in our own biology. Question about genetics, metabolism, encapsulation, and of course, is there or was there life outside the Earth in the solar system? And so I've been studying this for 40 years. My first work was as a high school intern at the University of Houston. I was studying the nature of prebiotic molecules. Then in college, my work was in biochemistry, doing enzymatic kinetics, but my senior thesis was on prebiotic chemistry. In graduate school, I was studying the nature of prebiotic materials and the first information macromolecules by trying to study the precursors of DNA and RNA that could have existed on the early Earth based on these chemical models that we can construct. I then did a postdoc at NASA Ames Research Center and the SETI Institute, where I was trying to synthesize plausible astrophysical ices based on what's seen in telescopes, but putting those in the laboratory and studying the compounds that are produced, the amino acids, as well as vesicles, encapsulation of molecules that we were able to see in these experiments. Over 20 years ago, I was hired at NASA Goddard Space Flight Center, and this was to further constrain the origin of life by looking at actual witnesses, as I showed in the first slide, looking at meteorites, the leftover pieces of the early Earth, of the early solar system, as well as looking at bits of the asteroid sample return mission OSIRIS-REx that collected samples from Bennu, which is what I've been working on for the last 20 years. Can I move forward? And so OSIRIS-REx collected material from the asteroid Bennu. It's a ancient primitive piece of the early solar system, and brought those samples back. These have been studied in the laboratory and are also available. Over 75% of the sample is untouched and available. We can look at the catalog and this QR code. This is a sample that's available at NASA, at Johnson Space Center. There's also samples at JAXA and soon at Canadian Space Agency. These are available for decades to come, so that scientists who have not yet been born can use techniques not yet invented to answer questions we haven't yet thought of. So anyone can write a proposal to look at these samples and study it with their own techniques, as I said, for decades to come. So far, we've found materials that could only be discovered in a pristine sample return mission. If they were in a meteorite, we wouldn't believe them, or they wouldn't survive because they're so fragile. We found an evaporitic condition where there are these minerals that dropped out, like in a dry lake bed, forming conditions that we believe would be very interesting, potentially plausible, the origin of life. It's a alkaline brine containing all the nucleobases found in DNA and RNA, as well as a number of other nitrogen heterocycles. 15 of the 20 amino acids found in biology, as well as a host of others. A wealth of ammonia, as well as other small molecules that indicate that this asteroid formed from a parent body in the outer solar system, rich with ammonia ices, that then travel to the inner solar system to get a sample of and bring it back to Earth. And so this is Jim Green, who at the time was director of planetary science, describing the NASA philosophy. Methodically, we work: fly by, orbit, land, rove, return samples. We've done that with the moon, and we're well on our way to doing that with Mars. There'll be other objects in the future we'll do the same process with. So it's getting closer and closer, stepwise, to getting samples on the Earth for places we can get to. And for places we can't get to, like outside of our solar system, or places like Oumuamua, we need to have the best analogs we can with the best data we can get from our observers that help us understand those. But there's a host of sample return missions, starting with Apollo 11, and then all the way up to, as I said, OSIRIS-REx that returned in 2023. And in development is the Artemis missions for NASA to send humans to the southern regions of the moon to collect samples, as well as the Japanese mission to go to Martian moon Phobos and collect samples. And then in development is the NASA ESA mission to collect samples from the moon and bring those back. But in the study of looking for life outside of the Earth, we may get lucky, like this joke cartoon. But most likely, we'll be looking for chemistry, not large organisms like mountain goats on Mars. And so, to connect these dots, we need to understand looking for biosignatures. But to do that, we need to understand the prebiotic chemistry from laboratory work and modeling, and how this overlaps with what is a biosignature, even for biology that we don't necessarily know about. That prebiotic chemistry overlaps with meteorites, which deliver prebiotic organic chemistry and other minerals to the Earth today that we can study as leftover bits of the solar system. But meteorites land on the ground, and they become instantly contaminated with both moisture and biology. So we need to understand their origin and the context for where these came from by looking at a primitive sample body like Bennu for the OSIRIS-REx mission. These are also need to be complemented by in situ missions, going like with the Mars rover to look at the location, the prebiotic chemistry, going with Europa Clipper, etc. Outer planet missions to look at these environments in situ. Although it's very difficult to detect a biosignature from orbit, there's a slight overlap. And all of this is necessary to get a sample, to know the geologic context, understand the exogenous chemistry, understand the endogenous chemistry. And that's necessary to understand how an evolved body can lead to sample return. And I think that's my time, but if it's not, then I'll show you the collection of the sample that OSIRIS-REx made on asteroid Bennu in 2020. Here is the sample arm descending to the surface to touch the asteroid within this 8-meter ellipse. This camera is the red square. This camera is the green square. And as the spacecraft is descending, it will make contact with the surface. You can see the shadow of the collection arm right here. It touches the surface. The spacecraft was descending at 10 centimeters a second. It actually penetrated, went underneath the surface by 0.5 meter, blew nitrogen gas to collect the sample, shooting up a cloud of dust and rocks as the spacecraft backed away to a safe location to then stow the sample, bring it back to Earth in 2023 for the world to study. Thank you.
Thank you, Dr. Dworkin. That video was well worth the extra couple minutes. Thank you. That's absolutely fascinating. Dr. Seager, you're up next.
Hi, everybody. I'm Professor Sara Seager at the Massachusetts Institute of Technology. How I got here, well, I always loved the night sky. You know, there's this saying that the sky is for everyone. And no matter where you are, even like me, I grew up in the inner city, there's always a few stars out there. And you can always kind of wonder what is out there. Well, when I got to undergrad, I was supposed to study all the sciences and go into pre-med and become the doctor that my family wished for. But I just loved astronomy, and I ended up being relatively good at physics, good at math, and just decided to pursue my passion. So long story short, I was a graduate student in the mid-1990s when exoplanets were first discovered. And at the time, they were really still very, very sketchy, like tiny signals. People didn't really, well, the signals were strong, but the kinds of planets astronomers started to find at the beginning were nothing like our solar system. And indeed, today, we have not truly found a solar system copy. So I'm going to tell you a bit about exoplanets, and then tell you about some of the key questions we have today. Here's a picture of the night sky. You can all see Orion there. Orion is setting now in the northern hemisphere. as spring finally arrives. Well, all these stars are suns, and if our sun has planet, it makes sense that other stars have planets also, and they do. And we astronomers have found thousands and thousands of planets. Literally, there will be trillions in our galaxy alone. But we're just kind of beginning to study those exoplanets, to study their atmospheres, to look for signs of life. You know how here on Earth, we have oxygen in our atmosphere that fills our atmosphere to 20% by volume. But without life, without plants and bacteria, we would have photosynthetic bacteria. We basically would have no oxygen at all. So literally, nearly 100 years ago, an astronomer, James Jeans, he actually considered this idea that because oxygen is so reactive, if we could see it on another planet, perhaps that would be a sign of life. So today, we are the first generation in human history that's trying to do this with exoplanets. And I do think I have enough time to squeeze in exactly how we're going to do it. Let's see if I can convey this to you. But we have some planets are very special. They are orbiting their star in the plane of the sky. Or in the plane, rather, of the sky let me say that again. Some of these exoplanet systems are very special. They orbit edge on so that from our viewpoint, the planet goes in front of the star as seen from our telescope. And in this cartoon here, you're seeing a cartoon. We do not spatially resolve stars like that other than our sun. But the artist is showing you this big planet going in front of the star, very exaggerated. And down here, you're seeing this, what we do see, a graph of the relative brightness of the star as a function of time. So most of the time, and the time is here in hours, the star is doing nothing. It's just shining at its regular brightness. But if we're lucky and there's an orbiting planet that happens to go in front of the star from our viewpoint, we see a tiny drop in brightness. And when the planet is finished transiting, it goes right back to where it was. So what I want you to take a look at here is seeing the artist's conception, what happens if there's an atmosphere. Now the planet is a tiny bit bigger. And one more piece of background is that when atmospheres absorb at different wavelengths or different colors, they absorb more strongly sometimes than others. We want to liken this to like a skunk spray, just the tiniest amount of that skunk spray smells really, really terrible. So some gases are very strongly absorbing at some wavelengths. So for a moment, I want you to imagine we're observing an exoplanet. We're looking at this so-called transit, when the drop in brightness happens, when the planet goes in front of the star, and we're looking at a wavelength where the atmosphere is fully transparent. Then we're going to see a drop in brightness corresponding to the dark part of this disk compared to the whole star. But if we look at a wavelength where the atmosphere is very strongly absorbing, all of a sudden that planet is a little bigger. Like if you didn't understand anything I said, it's just like this. The planet looks bigger at wavelengths where the atmosphere is strongly absorbing, making that entire planet look bigger. And now what I wanted you to do is look at some fantastic data from the James Webb Space Telescope. This is a planet called WASP-39b. And even if you didn't understand what I said, you can still try this exercise out yourself. We're seeing a transit light curve where the star is normalized to 1. So the star is just shining, doing its thing as the minutes go by. And at some point, the planet happens to go in front of our viewpoint and we see this tiny drop in brightness. And your job is to look at the different wavelengths or these colors and see where the planet looks a tiny bit bigger. You can type it out in the Slido if you want to. There's one wavelength where this planet is looking a tiny bit bigger than every other wavelength, and black is the best fit to the data. What you're seeing here is the planet atmosphere very strongly absorbing at the specific wavelength of about 4-point-something microns. And in exoplanets, we kind of take that and we put it on a diagram showing amount of light blocked as a function of the wavelength. And what you were looking at was this giant feature here on the far right. That is carbon dioxide on a giant exoplanet atmosphere. So, wow, I mean, that is cool. Hopefully, I mean, just with that data alone by eye, we could see the planet looks a tiny bit bigger at wavelengths where carbon dioxide is strongly absorbing. And if you didn't get any of that, let me just say that we're looking at the amount of light blocked as a function of wavelength. And for a planet with no atmosphere or no detectable atmosphere, the white dots would just be a straight line. Those are points from the James Webb Space Telescope. But because we're seeing this up, down, up, down, up, down, it is evidence of an exoplanet atmosphere on this giant planet with carbon dioxide and sulfur dioxide and water vapor and carbon monoxide and some other gases. And this is a giant planet that is too hot for life, but we're trying to push this down to smaller and smaller exoplanets so we can find signs of life by way of gases that don't belong. So I just wanted to say that there's way more astrochemistry than any astronomer ever expected, mostly because of photochemistry. Ultraviolet radiation from the star breaks up molecules and will set off a chain of events, reconfiguring some molecules and making them very different from what we initially expected in our simple chemistry atmosphere codes. Well, that little fast description was just to get you to believe and to agree that we do have data. We're studying exoplanet atmospheres. We're trying to get down to smaller and smaller exoplanets. We're asking ourselves, what are all the different gases that might be signs of life? It's not just oxygen, but think of that mold in your fridge or your compost that smells really bad. That is a gas made by life. When you walk in the pine forests and all the smells coming off trees, made by life. And we literally have a very long list of gases right now. We're getting ready to interpret anything unusual we might see. But in going through this exercise of thinking of all the different gases, I came across my favorite gas called phosphine. Now, some of you may have heard of phosphine, those people who follow space science, that 5 years ago, I was part of a team where we made an announcement about the detection of phosphine gas in the Venus atmosphere. This remains highly controversial. It's a tiny signal in what's very noisy data. But here's the thing is that we followed this kind of paradigm I brought up in the beginning, that there's a gas that shouldn't be there. It's highly reactive. It doesn't seem to be made by volcanoes or lightning or meteor delivery. And we made this announcement to the world. And this discovery was pushed back. For lack of a better word, let me just say non-scientific parlance, trashed at every level. And I'm just putting this out here for a moment because finding gases on exoplanets is tricky. If you look here, we have these squiggles. We're trying to call them different gases. We can usually identify that a gas is present, but it's very hard for us to say exactly how much of that gas is actually there. And there's a lot of challenges in looking for tiny signals, not only on Venus, but on exoplanets, of course. So this is highly controversial, and it gives us pause about how we're going to find signs of life on exoplanets. And that's one of the biggest challenges we have right now. So let me just say a word about Venus for a moment, because in my career, I'd worked on exoplanets for a very long time with this goal of finding signs of life. And this controversial discovery of phosphine on Venus, it sent my research in a completely different direction. And I'm just going to summarize that briefly for you, saying that, Venus is too hot for life of any kind at its surface. But just like on Earth, above the surface, it gets colder and colder until about 50 kilometers above, it is the right temperature for life. And that's why half a century ago, Carl Sagan put out the idea that maybe there's life in the Venus clouds. Our Earth clouds have some temporary life in them, bacteria. Maybe the Venus clouds do also. But it's not clear if at the time they understood that the clouds of Venus are not made of water. They're made of concentrated sulfuric acid, a nasty toxic chemical that destroys our DNA, destroys all life on Earth. But now I have a new branch of astrochemistry to tell you about. And that is work that my team has pursued ever since this reported discovery of phosphine on Venus, that we have been experimenting in the laboratory with concentrated sulfuric acid. And we have shown that our 20 amino acids are stable. We have shown that PNA, peptide nucleic acid, is stable up to 50 degrees Celsius. We have so many results, I couldn't possibly summarize them all for you. But I wanted you to know that we are literally pushing new exploration about sulfuric acid itself as a potential solvent for life. Tied together with this, we're trying to go back to Venus with a series of small focused missions to search for signs of life, and hopefully one day, connecting to Jason's talk, a sample return from the Venus atmosphere. We have our first mission to Venus. You can just look it up, Google it, Rocket Lab mission to Venus. We just got back from Hawaii to test our instrument on a drone, like a light aircraft flying through some volcanic fog. This was credit to Professor Chris Carr from Georgia Tech. And we have other missions planned as well to go to the Venus atmosphere and search for signs of life. I may be getting a bit ahead of ourselves, but so many things are happening and we're super excited about the search for life beyond Earth, but we've run into a lot of bottlenecks. And I think during the Q&A, we can talk about it. So I just wanted to put up some ideas of where we're going to be in 2050. It's kind of a wish list where we need to be if we're going to really nail finding signs of life beyond Earth. Thank you.
Thank you, Dr. Seager. I've got a lot of follow-ups about Venus in particular, but we'll save those for the Q&A right now. Let's turn it over to Dr. Talley to wrap up the initial presentations.
All right, thank you, Ed. My name is Jennifer Talley, and my journey to where I am in this field is perhaps a bit tortured, and I'll try to make it short. So I have always loved mathematics and the ability of math to both explain and predict everything we see around us in our world, and connecting that to biology. For me, both in my education and then professionally, I've tried to connect that back in a cycle, that if we can use math to explain and predict everything around us, especially these biological systems and solutions, then how do we use that those biology solutions with our better understanding to improve our technologies? And so I started off and for several years worked in understanding specifically how the nervous system of different organisms processed information, either optically or chemically, was able to take in that information, process it in the context for that organism, and then make a decision about what needed to be done and enact that. And so that was my work for the Air Force for more than a decade, was really these basic fundamental questions about how biology does what it does really well, and then how can we work with engineers to improve our technology based on that. And then with the Air Force and the Space Force sending me to Air War College, and I'm moving me into more of a strategic position here with AFOSR. What I've shifted is more to asking, what basic research do we need to invest in now for our future needs? And I've been put in charge of what's called space biosciences. So I think, unlike everyone else on the panel, I've really only been thinking about the space environment professionally for not quite 2 years. But in a way, it is continuing what I've always been passionate about. And in listening to the other panel members, it's kind of putting together what they're studying to improve our technology for the Air Force and the Space Force. And so Dr. Bergner talked about these extreme conditions of space. And if you combine that with Dr. Dworkin's work, understanding genetics and metabolic pathways and how those came about, with Dr. Seager's work in looking for signs of life out in these conditions and what that may be like, what I'm trying to do is fund basic research that answers the question of what basic science questions do we need to understand in order to improve our technology in space, and what is the role for biology in that? And so I look at the extremes of space as both a challenge for life and for biology, and as an opportunity, because what biology can do really well, looking at things like polyextremophiles, these microbes that can over selection evolve to handle incredibly challenging resource-constrained conditions and meet those needs. And I want to understand, what are the genetics, what are the metabolic pathways underlying those that could provide us with better materials for the space environment, for these extreme conditions, and could we actually have living systems in space to kind of meet these needs. And so that's the research area that I am moving into. And I would say that some of the discoveries recently have been the feasibility of biomanufacturing in space. So there's a few studies looking at how we can have engineered microbes produce things in the space environment for our needs. So, the feasibility has been shown out, but it actually opened up even more of these fundamental research questions. And there's a question of what we can synthetically engineer into these microbes, into these natural biological systems and materials for the space domain versus what already naturally occurs. What can we learn from polyextremophiles? And first, we have to understand them, how they do what they do, before we can actually use them to improve our technologies. And so I think some of the big challenges for this field going forward include things like synthetic biology, design, build, test, learn cycle. So if you want to produce a synthetic biological organism that produces something here terrestrially, I have an example of a company that spent 5 years and about $20 million to make one acid compound here terrestrially. When you add in spaceflight to that, the cost and the time go up exponentially. So we need to find new rules, new tools to do better prediction of what experiments we actually should send to space to test out. And why do we need to send to space? Because the conditions in space we cannot reproduce in all ways here on Earth. And that has, I think, as you've heard, impacts on the chemistry and therefore the biology of these systems. In addition, another challenge I see for my area is, what is homeostasis? If we have life in these extreme space conditions, either a living material or a living system that's doing biomanufacturing, what does homeostasis look like? And what are these fundamental basic research questions that we need to answer to get at that? And I think, what would this field look like in 2050 depends a lot on what our commercial low Earth orbit destinations look like. So there's consulting companies that project the future space economy to be about $1.8 trillion by 2030. So that's just in 5 years. In 2030, the International Space Station is projected to be decommissioned and deorbited. So we're going to be losing access in a lot of ways to space experiments. And so I think what this field looks like depends a lot on the commercial access and the price to go to space. There are several companies trying to put low Earth orbit destinations into space, but we haven't yet shown and proven out a test commercial use case, right, that would actually be profitable and sustain these destinations. I think there's a lot of excitement, there's a lot of activity, but we're kind of waiting for that. And so I just want to end by saying that I think it's incredible space to work in, speaking of dumb jokes, and an incredible opportunity to see where this world is going to go. And I think, being at AFOSR is this incredible place where, our mission truly is to invest in basic research science. And then, of course, with one eye always to serving the Air Force and the Space Force mission. And it's really exciting that I get to work with and then leverage and use a lot of the great chemistry and biology that's going on as you've heard today. Thank you. Over.
Thank you, Dr. Talley. And thank you all to all the panelists for those just absolutely fascinating presentations. I think we can go into some questions right now that I had, and then we'll get into audience questions after that. My first question, as I sort of intimated earlier, is for Dr. Seager, related to Venus specifically. I followed the news when the phosphine discovery came out a few years ago. I thought it was absolutely fascinating. Venus is a really interesting target in terms of search for life. My understanding, you can correct me if I'm wrong about this, which I very well might be, is that we believe Venus may have been quite a bit more Earth-like up until maybe 700 or 800 million years ago, when there was sort of a disastrous resurfacing event that resulted in the very Go ahead.
Hi, can you hear me okay?
Yeah.
Well, Venus is interesting because it seems to be controversial on every topic. There are a group of, I thought that most people believe Venus formed like Earth, that it had oceans and it was very nice and life may have formed. And one idea is that as Venus got hotter and hotter, it was close to our sun, but there was an event, like a massive volcanic event that made its atmosphere even warmer, pushed it over the edge, it lost its oceans and became the planet it is today. And perhaps if there was life early on, it migrated to the clouds and changed. But other work hypothesizes that Venus was always too warm for water to be condensed in its liquid form. And it's because of the atmosphere and clouds and just the whole energy budget overall. So it's still controversial as to what Venus was early on. And we're hoping that the next set of missions to Venus, namely NASA's Da Vinci and other missions, will look for minerals on the surface and do more detailed studies to help us sort through those two options. But was your question about life or just about the early Venus?
You anticipated my question. That's exactly where I was going. In terms of life in the clouds, the question would be exactly as you just stated. Is it reasonable to think that life might have formed on the ground in the same way that it did on Earth in a more benign environment some hundreds of millions of billions of years ago, and then seeded the clouds and maybe survived there, persisted there after the surface became completely untenable? So it sounds like your statement is that that's controversial at this point.
One theme I didn't quite get out there in my talk was like early on, I started on exoplanets, super controversial, super crazy, all mainstream now. Now I'm working on Venus, this whole idea of life, super controversial, super crazy. It's not really accepted as legit in many ways. But what I'm trying to say is not that we have to work out every theory, do all of our detailed chemistry. I'm saying, hey, let's go and check it out. Let's bring our new miniaturized sensors. Let's see what's in these cloud particles. Let's look for complex organics. We just heard from Dr. Jason Dworkin about, wow, all those amazing things in meteorites that could be delivered to Venus even. So let's go there.
So that actually leads me to a broader question that I'd like to sort of go around the horn with for all the panelists. When we talk about life on exoplanets, we often talk about the Goldilocks zone, right? And planets which are sort of in the same general range that the Earth is in terms of insolation and expected surface temperature. So we're sort of assuming water-based chemistry. And my question is, do we feel that we are being overly narrow in doing that? Do we feel that there other, as you sort of intimated, Dr. Seager, do we feel that there are other bases potentially for life? And then follow-on question to that, if the answer to that is yes, how would we detect life of that sort that uses a fundamentally different chemistry than the life that we have here? Because we know, more or less what the biosigns are for sort of a carbon, nitrogen, oxygen-based life with using water as a solvent. Do we know what the chemical signatures would be for a type of life that was using a more alien chemistry? And maybe we can just start from the top and start with Dr. Bergner. It was your presentation that made me first think about this. So what are your thoughts?
Yeah, I mean, I think that organic life has a lot going for it. We know that water is very abundant in the universe and available in regions where planet formation is taking place. And we know that carbon, oxygen, nitrogen, these organic building blocks are also abundant and even present in chemically complex forms in environments where planets are forming. From a cosmic abundance perspective also, you have a lot more carbon, nitrogen, and oxygen to work with than something like silicate. So I'm of the opinion that the low-hanging fruit is to look for organic life. Not to say that it can't exist in other forms. I'm not an expert on other forms of prebiotic chemistry, but astrophysically, there's no reason not to expect that these building blocks are more or less ubiquitous.
Okay, thank you. Dr. Dworkin, you have any thoughts on this?
Absolutely. So, as I said, water is a good bet, but there's other possibilities as well. We heard about sulfuric acid, liquid hydrocarbons, formamide, all kinds of things could happen. What to look for are chemistry that's out of equilibrium, something that is showing an active process that doesn't look like geology, that doesn't look like meteoritic infall. Now, again, water is a good bet, and so looking at solar system, at star systems that have a habitable zone that has water is a good idea. But of course, this habitable zone is not just distance from the sun. There are microenvironments on objects like the Earth, where, like the top of Mount Everest, water isn't so stable as liquid, and so there are moons, there are altitudes, there are subsurface locations outside of the classic habitable zone that could also contain a clement environment for life. We don't know why life forms and how it forms. So one of the mysteries that I have with asteroid Bennu is that it is this clement environment, liquid water, mildly alkaline pH, salts, organics, but no evidence of life. What did Bennu not have that Earth had? That's a rich area for experimentation in the future.
Okay. Dr. Seager, you seem to be more a fan of alternative chemistries. Maybe I'm wrong about that, but what are your opinions here?
Well, I definitely like being contrary, but this time I have to be boring and agree with everyone. Organics. We want to look for complex organics that are not coming, like we just heard, from asteroids, or just wouldn't expect to be there on their own. Now, what exactly those are, where do we draw the line between what will be there on its own and what is so complex it has to be associated with life? We're not sure yet, but complex organics.
Okay, Dr. Talley, what about you? Any thoughts here?
More that I'd be really interested in if things could be non-organic, right? Because in the usage of space for humans, for our technology, is really dependent on life as we know it, right? And so, yeah, 90-something% of the water on the International Space Station is recycled. But if we think about this future space economy that they're projecting to be, all resources are going to be heavily constrained in space. We already know that, right? And so thinking about what can we use out there if we're sticking to the chemistry that we know and trying to reproduce terrestrially what we have here, that may be that's definitely one way to go, and it's the way that we're currently looking at it. But should we look at it from, how else can we support life with alternative chemistries would be really fascinating to me.
Okay, so it seems like the general consensus is that sort of conventional organic life is our best bet at this point. So let's talk a little bit about the idea that Dr. Dworkin raised about microenvironments. Again, looking at spaces outside of the sort of classic habitable zone. I think there's some evidence, maybe very strong evidence, that we have liquid water subsurface on some of the moons of the gas giants. Obviously, you have the issue there with energy, providing energy for life. Anything that was occurring in those subsurface oceans would not have access to solar radiation, but might have access to alternative chemical sources of energy for the origin of life or sustenance of life. So again, just sort of going around, starting with Dr. Dworkin, since you kind of brought this up. Any thoughts on the possibility of life and organic chemistry occurring in those sorts of environments?
Certainly. The problem is we have one example of life, because all biology on Earth is connected. It shares the same chemistry, the same language, biochemical language, and so we're extrapolating off of one point, which is hard. We also know that on Earth, everywhere you look, you find biology. So if you have a robust ecosystem, it's widespread. One of my fears is looking at a place that has a very small biological system. So if you imagine, Enceladus or what have you, that has a lot of organic chemistry, and how would you detect one cell that's eking out a living barely amidst all of that other background. And that, I don't know how to solve that problem, especially when you don't know what life you're looking for is. But there are absolutely lots of possible niches, and it's limited by both our imagination and our understanding of chemistry.
Right. Dr. Seager, in terms of microenvironments, you brought up again the clouds of Venus. My understanding, and I think you mentioned this, that you said roughly 50 kilometers, you get a temperature around 70 degrees, a sort of a nice afternoon, and temperature relatively close, or pressure relatively close to what we see on Earth, but not such a great chemical environment. So you said just now that you thought organic, sort of traditional organic chemistry is our best bet, but that would not be viable in that environment, would it? And if not, what sort of life do you think would be viable there?
No, definitely viable. We have shown that complex molecules can survive. I mean, it doesn't mean there's life there, but our extended team has shown that you can start with formaldehyde or little organic molecule, and in sulfuric acid, it will grow into a rich organic chemistry, to aromatics, to ringed molecules. And this was actually, oddly enough, well known in the oil industry, not related to planetary science at all, that complex molecules could form. I mean, we have shown that lipids with a polar head, if you put those in sulfuric acid, just like some of them in water, they will point their heads out and make a little tiny vesicle. It's not like a cell membrane, but think of like the most primitive vesicle you could imagine. And people had done this work decades ago. It just wasn't known, actually. So they can survive. We're trying to make a DNA-like molecule, not our DNA, but swapping out parts. And we recently had a big milestone. In the end, no one cared about it, it turned out, but we have shown PNA, peptide nucleic acid. So our nucleic acid bases attached to what's like a glycine backbone actually is stable in concentrated sulfuric acid, not to high enough temperatures, well, higher than room temperature for sure, but not extending all the temperatures. So I think we're getting to the point, we have way more to do. There's way more unpublished work that I think I can convince you, if I have more time, that there are complex organics, whether or not they're life-based, whether or not there is life is separate. So yes, we are in phase A, literally, of a mission to go to Venus. That will be a balloon, like the balloons, but more sophisticated, that went in the late 1970s from the former Soviet Union. We have a mass spectrometer that already exists that will look for these complex molecules. We are developing how we're going to collect the cloud particles, condense them, evaporate most of the sulfuric acid. We are working all that. We are in phase A. And the only thing we have left that's super hard that we're starting this summer, I'm like just repeating what Dr. Jason Dworkin keeps saying, is how are we going to recognize what we're looking for? And we do have a team this summer trying to work on that. Okay, because like mostly when you're using mass spec here on Earth, you've synthesized some kind of chemical and you want to verify what you found. But we're doing like this blind search of like, wow, we're going to like put stuff in our mass spec, and how are we going to know it's there? So I just went a little overboard in answering your question, only because I'm so enthused about this, about the evidence that there are organic molecules, the mission we want to send, and just the hard part in uncovering what's there.
No, overboard is great. We're all about overboard here. No worries with that. Dr. Talley, your thoughts on this, if any?
Yeah, back to I do care, Dr. Seager, a lot, actually. That sounds really interesting. How can we, so for me, whether or not it can sustain life, whether it is classified as life, doesn't matter as much, because I want to use it. And so can we make a multi-use material that has some better property to it, from something that is organic, but not necessarily life. To me, that's the interesting part. But that comes, again, along with a lot of fundamental questions about what conditions they can survive in, under what conditions they can form, what are their properties in that environment that would need to be answered.
Hey, thank you. And Dr. Bergner, background to you. You seem, pretty set on the sort of water, carbon, nitrogen, sort of traditional chemistry. What are your thoughts on that occurring outside of what we would consider the traditional habitable zone?
Yeah, I think it's definitely interesting to think about, and obviously the environment that life is emerging in will dramatically shape the outcomes. So during this discussion, I was reminded of a study I saw a while ago showing that the nucleobases used by life on Earth are sort of preferentially photostable to UV radiation received at the surface of our planet. And so I think that even if you start with all the same sort of building blocks and chemical mechanisms, the way that the life develops may be very different if your evolutionary pressures are in the clouds of Venus compared to the surface of Earth. So I think this is all really interesting to think about.
Okay, so going back around to start again, Dr. Bergner, since we've got you up on the screen already. This seminar is Chemistry 2050. So what advances in technology do you anticipate might occur in the next 25 years, or do you hope will occur in the next 25 years that could help advance your work specifically?
Yeah, so I think on the sort of telescope facility side, there are a lot of exciting things in the pipeline. I mentioned the possibility for a far infrared mission. There are also a number of other facilities with different wavelength coverage that are being discussed. And the really fantastic thing about working with telescopes is that every time we get a new one, it's sort of a paradigm shift in the field. And so sort of regardless of which telescopes end up being chosen, I think we can anticipate a lot of new science enabled by that. I also think that as our facilities get fancier, as we get more data, that more advanced computing and machine learning is going to play an increasingly important role in sort of parsing what we're seeing. Maybe there's a role for that in what Sarah and Jason were discussing as well, of if you're sort of swamped by information, how do you pull out meaningful trends from that? And so I think probably across the sciences in general, we're going to be seeing a much larger role of AI and machine learning in science. But I can certainly imagine that applied to our community in astrochemistry as well.
Thank you. Dr. Dworkin, same question to you. What advances do you think might occur in the next 25 years in technology that can help advance your work?
Sure. Well, let me first sidetrack and say that, look, there's a lot of stratospheric sulfuric acid on Earth. And I would expect that to be a niche colonized by terrestrial biology if they can figure out how to do it. So I'd send a aircraft that's been decontaminated to take a look. That's going to be a tricky task, but that would be very interesting to look for the sorts of organic chemistry that can be seen there. I know that actually micrometeorites form nucleation sites that sulfuric acid droplets form around. And that also gets some astrochemical information mixed into the sulfuric acid. So that's another confusing factor to look at. But I couldn't hold that in. I'm sure Sarah knows about that. But so more samples, more techniques that can look at smaller and smaller samples. We've had great breakthroughs where, for example, the Stardust mission brought back dust from the coma of Comet Field 2, and techniques were invented for that where a single micron grain was too large to be analyzed. It had to be sliced up into smaller and smaller pieces to be looked at for elemental and isotopic composition. But we need that same approach for organic chemistry to get the names of compounds, their chirality, obviously their structures, from small particles in situ. The way we do it now is extracting large amounts, milligrams, in solvent or hot water to pull the organic compounds out like steeping tea. That's inefficient, and that also alters the chemistry. Now, that alteration is similar to what would happen if it landed in an ocean on the early Earth or where have you. So it doesn't affect the outcome, but knowing the astrochemistry, that's altered and that's diluted. So the ability to look grain to grain, the chemical structures of the organic compounds would be fantastic. Right now, we can only do that by formulas. which is great, but not enough. And by isotopic regions. Again, great, but not enough. Need more of that. And of course, more samples from Mars, the Moon, Ceres, Europa, and Enceladus everywhere.
Hey, thank you. Dr. Seager, what about you? Any technologies you're looking forward to advance your work in the next 25 years?
Well, I have a few actually. One I've really started trying to get my head around. We call it ab initio methods, like quantum. Like in my computer, because we're doing these sulfuric acid-based experiments and some other solvents, but you kind of run out of time. Like, got to get a molecule to synthesize. That takes a while. Put it in sulfuric acid. Okay, it fails. Start again. Blah blah blah. But if you can get some guidance from your computer. But the problem is when we want to do these calculations, it's really about quantum mechanics. That's okay. I think all physics is chemistry at the end of the day. I mean, at least when it comes to molecules. And basically, it's so complicated, but I'd like that tool to be more easy to use, to have faster computers. Maybe the quantum computers will be working eventually, and we can put a quantum problem on a quantum computer. So that's one thing. Secondly, it's related to what Jason just said, but I'm super into photonics. You know, they're these little tiny chips and you send light through these little tiny, it's just incredible that you can shrink giant tabletop things down to like this big. And I've been really keeping my eye open for ways that chemistry can help with planetary missions. For lack of a better description, think of confetti. Like we go to a planet and it's really bad to litter, but throw a bunch of like little chips all around and search for that elusive molecule or set of molecules we're looking for. And I don't know, like there's going to be a lot of work to get this ready, but all the ingredients are out there. There's some complicated, more power hungry ones, like they're called frequency combs, where you tune it in to match a certain molecules, like infrared spectrum. There are these really cool new, I don't know how new it is, it's new to me, carbon nanotubes. These tubes and the aromatic rings, like they're really easy to fluoresce. And you put on like an add-on to it for the molecule or the class of molecules you're looking for. So when that in the environment matches onto the molecule you're looking for, it changes the way that this molecule fluoresces actually. If we could combine that with photonics, wow. So I'm just giving you some specific examples, but I just think this like jump in miniaturization for our sensing methods is going to be a major game changer. And I do want to say it's hopeful, but realistic that could be a new way to do planetary science in the future in 2050.
Okay, great. Dr. Talley, what about you? Any thoughts?
Yeah, so much. Kind of touches on a little bit of what's mentioned already, that, you know, this AI and machine learning predictive methods, right, could become very powerful. AlphaFold 2 is a program, large language type AI model that predicts the form of a protein given the sequence. What we don't have is form to function, and we don't have multiple combined functions to metabolic pathways to whole cell behavior, right? So speaking to the complexity, you know, of chemistry, the complexity of that interaction with physics and the chemistry underlying that, you have a huge parameter space when you try to do, you know, the prediction of a, you know, biological experiment. And then when you are adding in, you know, genetic manipulations to that, right, then that becomes exponentially more difficult. So I'm looking forward to more predictive elements and this computer prediction for what experiments should we do out of this giant parameter space. Now, we need a lot more data to train those models as they currently exist. You know, getting data in some senses is a really boring project. I hear from, you know, professors all the time that say they can't get those types of projects funded. So I think a little bit of shift in the funding agencies, you know, to look at these projects and the people who want to go after these large data sets that we really need. Maybe new, that what's the next AI tool that maybe doesn't depend on these large training data sets? Can we go back to the, you know, the neural networks and figure out how biology, you know, does prediction without these large training data sets? I touched on the commercial low Earth orbit destinations. So the International Space Station has been fully tasked since 2023. It's very difficult to get an experiment manifested both in terms of the physical space, on launch, on station, but also crew time to do those experiments. So automation, which is very, very different from autonomy, from AI, from machine learning, but just automation, so robotics in space. I'm really looking forward to the advantage that that could provide in easing some of that crew time, whether that's on the ISS, whether that's on other low Earth orbit destinations. And then I'm really looking forward to advances in sort of terrestrially space analogs, right? So NASA and a lot of the national labs have great facilities to do different types of radiation testing, but almost no one does these combinations of radiation and another condition, let alone all of them, right? So what I'd love to see is what I call the space box. Give me a box here on Earth that replicates everything about the space environment that I can tune up and down. It can't exist. We can't make microgravity. We can't make anti-gravity. It violates the laws of physics. The ways that we simulate microgravity are not great for biology. In some ways, they seem to be okay, but even sussing out how and where they work well to simulate that and how they don't is difficult and expensive and time consuming. So I'm really looking forward to these combinatorial better analogs for the space environment as well. And then to the tools that we actually want to use. So CRISPR-Cas9, another Nobel-winning advancement in my field, incredible tool for doing these genetic manipulations. So, what's the next generation of that will help us do this faster and with less expense?
Okay, great. So this is a tougher question. As long as we've got you up on screen, we'll just start with you. And if the answer is just a big shrug, that's okay too. But can you anticipate, or are you aware right now of any major blind spots in the current research environment, things that should be investigated, avenues that should be pursued that are just not being pursued by the current funders, by the current academic world, or research world?
Yeah, I mean, I think the large data sets, that's definitely a hole. Biology is complex, and I don't think that we have enough data to really understand the complexity in a lot of cases. And so I think data is something that people just are not willing to put funding towards, because it's not as sexy, it's not as exciting. Other than that, we don't know what we don't know, right? So actually the military spends a lot of time doing what they call future forecasting, where they sit down and try to figure out what the future looks like, and you're almost always wrong. So it's pretty exciting when something comes along that really changes the way that we can do science and the way that we can think about science. So I'm looking forward to those gaps, but I would be just as bad as everyone else in predicting what they actually are.
Fair enough. Dr. Bergner, what about you? What are we missing?
Yeah, maybe related to what Jennifer was mentioning, I think it's often I think it's well recognized actually that lab astrophysics is underfunded compared to the needs of the community. Again, maybe it's not the most exciting science to be measuring like fundamental optical properties of materials or reaction kinetics for thousands of different reactions, but this is so important to the way that we interpret our data, and all of our conclusions are built off of these fundamental studies. So I think higher prioritization for some of the fundamental work is really important. And why will we build billions of dollars of telescopes if we can't actually interpret the results from them? So I think that's a big area. Let's see, other bottlenecks. I think that's the biggest one. I will say for something that sort of intrigues me in general about astrochemistry is the span of size scales and time scales. So we have to grow from, molecules are sort of angstrom nanometer scale. We have dust particles that are submicron scale, and we need to build planets that are tens of kilometers scale, or thousands of kilometers, sorry. And so having sort of frameworks for thinking about all the processes going on across, I don't know, 17 orders of magnitude and size scales. And then again, chemical processes are happening on incredibly fast time scales, maybe picoseconds or faster. And then planet formation is happening on million-year time scales. And so how do we sort of wrap our heads and develop models that can accommodate these huge spans of physical size and time, I think is the major fundamental challenge.
Thank you. Dr. Dworkin, what about you? What are we missing in the world right now?
So as Jennifer and Jenny just said, we have issues with the what do you call it? Low risk, low excitement experiments, that the fundamental constants that we need to put into these machine learning models, so that they can understand the astrochemistry of the billions of different possible organic compounds, not just the 90 some elements on the periodic table, and how they interact, but also the high risk, high consequence experiments, like some of them that Sara was describing, looking at alternative chemistries that could have impacts, and getting both of those funded can be difficult, but also doing things which are exceptionally expensive, and anything involving spaceflight is in that category. So anything that can lower the barriers of entry into space, either by reducing the mass of a payload, increasing the reliability of a robotic or detector device, and of course reducing the cost of getting into space and out of Earth orbit, are all limiting factors on the things we can do.
Thank you. Dr. Seager, what about you? What's not getting funded that should?
I'd love to see more small space missions get funded. It's a bit of an oxymoron because, yeah, everything's super expensive in space, but we have traditionally had so many amazing missions. The asteroid sample return from Bennu was incredible, all the Mars rovers, but it would be really nice to not wait like decades to go back to Venus or some of the other things, and to try to take as much advantage as we possibly can from what we call like the commercial spaceflight world, where there are smaller, faster, more nimble opportunities. And I don't think that we have really, as a community, really grabbed onto that.
Okay, thank you. All right, we've got about 30 minutes left, give or take, maybe a little less. So let's go to questions from the audience now. I'm going to try to direct these to the panelist who I think maybe is best situated on first cut. I could be completely wrong about who is best situated for any of these questions, though. So once that person has answered, if anybody else wants to jump in, please do so at that time. So first question, and I'm going to direct this to Dr. Talley. One of the major limitations of building larger space telescopes is getting larger mirrors into orbit. What creative options do we have for implementing this in future missions?
Yeah, that's an interesting one. So creative options. All right, creative options. So we could grow things in space, right? Biology loves to eat things, break them down, and create new compounds. So if space activity commercially and for tourists is going to be in our future as a species, we're going to have a lot of people and activity in space. What does manufacturing in orbit look like? So what can we not only manufacture in orbit, but what can we recycle? What can we grow? What can we make? What's the role for biology in that? I also think taking lessons from some of these biological solutions for space. I've seen, and I think that they are using some concepts from folding structures, things that change shape and adapt. And, things don't have to be perfectly aligned if your structure can learn from its own shape, its own performance, and adapt shape. I don't know if anyone else on the panel has thoughts on that, but.
Anyone else want to weigh in on this? Okay, fair enough. Next question. I'm going to direct this initially to Dr. Dworkin, but again, after he has a chance to take a crack at it, anybody else can certainly weigh in. The Europa Clipper mission is the state of the art on in situ astrochemistry. However, NASA Neptune orbiter proposals include the older ion and neutral mass spectrometer instrument used in the Cassini mission. What advancements are needed in such sensors for probes?
So everything's about risk and about minimizing your risk, because getting to space is expensive. And so you don't want to put a spacecraft that has a high probability of failure, or instruments that have high probability of failure if the launch is so expensive. And so there's something that's used in the business called TRL, technical readiness level, that you have to have an instrument that has been demonstrated to work under these environments. And so the more expensive it is to get a place, the more conservative you want to be about making sure that your payload works. Furthermore, the harder it is to get to a place, the more energy it takes, and therefore the more expensive it is to fly something with a lot of mass. So if you want to send a CubeSat to the outer planets, great, but it won't have the pointing stability you might want to focus your instruments at a small target or a moving target. It won't have the bandwidth to send data back as far as Earth, and some things. And if you can, there have been cases where off-the-shelf equipment has been used on satellites, on small spacecraft, but there's a very high failure rate. And unless we can accept faster, better, and cheaper, all three of them, that means accepting a higher failure rate. And as a country, we're not willing to do that. Things go bad. So the more expensive the mission, the more conservative you need to be to make sure it doesn't fail. And you don't have to have your PI or administrator stand before Congress to explain why the mission didn't work.
Okay, appreciate that. Anybody else want to weigh in on this one? Okay, this next question is directed to Dr. Bergner. You mentioned in your presentation the exotic weirdness of astrochemistry. What kinds of exotic molecules do we expect to find in space?
Yeah, great question. So I guess one of the classes of molecules that first struck me is you have these really large, very unsaturated carbon chain molecules. So HC7N, HC11N, these are 11 carbons stuck in a row with hydrogen on one end and nitrogen on the other. So these are the kinds of molecules that you would never see on Earth, and it's kind of surprising to find out that they exist. And then on the other hand, we have sort of exotic phases of matter. So I mentioned the interstellar ices. This is sort of a fluffy, amorphous phase of solid material that is different from anything that we see here on Earth. And so while the molecules that are making up these ices are familiar to us, the sort of form and microphysics of that material is very different.
Thank you. All right, this one's back to Dr. Dworkin. During your presentation, you showed a joke about mountain goats on Mars. While this was a joke, it does bring up a serious question. If we focus on sample return and in situ analysis missions, we may miss the mountain goats if they are there. But remote sensing won't be good enough to see the mountain goats if they're outside our solar system. How can we make sure we don't miss anything?
Sure. Let me also put in that with the exotic molecules Jenny was talking about, there's also an intrinsic selection bias, at least for some techniques. So these very long molecules, HC7N, are really easy to detect with rotational spectroscopy. And so they jump out at you. So they may be a very rare phase, but they're easy to see. That's the key under the lamppost issue. While other phases that maybe are common are harder to see, like N2, for example. But that they exist in space at all is still exciting because there's collisions are rare. But I go back to a comment that Carl Sagan made at one point about a similar question about detecting something weird. And he referred to this as a silicon giraffe. That if a silicon giraffe walked in front of the Viking lander, you'd see it because you've got a camera. And so all missions have cameras. You would see something that's macroscopic and big. Something that's abundant would impact the environment. On a satellite level, you can see forestation or things of that sort. But there's all these critters also would emit compounds that you're looking for, the same ones who are looking for microbes. Big critters also emit compounds that are out of equilibrium with their environments. They also emit methane, maybe phosphine. Depends on the kind of chemistry that they have. But as we've said, missions are expensive. Even ground-based telescopes are expensive. And so you need to only look for what you have a probability of detecting, plus some phase space on either side. And so within a realistic budget, you need to focus on things that have a high probability of detecting. Again, the key on the lamppost analogy. So the more preliminary work that's done in the laboratory, in ground-based telescopes, the more we can understand the environment and the better we can make predictions about what we should be focusing on. But again, if they're go-to, see them.
I just
Okay.
want to add something here that if we
Yeah,
want
go
to,
ahead.
if we want to switch for a moment to exoplanets, where we don't have the kind of quality data we're going to get from visiting our solar system objects directly, we have quantity. Lots of exoplanets hopefully will be amenable to observations, and there we wouldn't be able to see not only the silicon giraffe, but anything really. And so we're trying to be positive, but at the same time waiting to see how things play out as people are starting to claim tentative signs of life on exoplanets with very noisy data and no real way to back it up. It's definitely a problem.
Absolutely. And so the best we can do is, of course, bigger and better telescopes, which just takes time and is expensive. Laboratory experiments that simulate these environments to do some of the fundamental chemistry that in the extreme environments to see what the signals look like, as well as looking at the exoplanets in our own backyard. The places like the ice giants and the gas giants and Venus that have maybe similar environments that we can then study to understand the exoplanets.
Yeah, like for example, I just got back from Hawaii, as I was mentioning, for the instrument testing, and the volcano was erupting, Kilauea on the Big Island. It was incredibly powerful. It was like 700 feet tall, like a giant fountain, giving off tons of gases. And what we're discussing here is, what if the exoplanet has just a slightly different amount of things on the inside? You know, that when the planets are born hot and we think they all have magma oceans, like oceans of rock, melted rock. But there's like a special time scale as the atmosphere is evolving and the initial inventory. And as soon as that turns into rock, stuff's like literally frozen in and later can come out in volcanoes. So all of that is unknown for exoplanets, but there are laboratories starting to simulate, like literally not simulate, like do real experiments in like tiny little crucibles, like imagine volcanoes and materials elsewhere. So yeah, we could definitely do a lot of work trying to figure things out. But it's tough for us to like expand our minds to all the possibilities out there. Like this favorite molecule, DMS, dimethyl sulfide, it's been found in a comet. But people want to associate it with life on an exoplanet. So it's kind of, how do we handle all those gases that are in minute quantities here on Earth, and whether or not they're always going to be life dominated?
So that leads us well into our next question, which is directed to you, Dr. Seager, and you may have already partially answered this. You noted in your presentation that we are the first generation with the ability to observe exoplanets. If you could build any telescope to study exoplanets, what types of planets would you observe and what chemistry would you want to study?
Well, I would actually like to be able to find an Earth twin, a very reliable scenario where it's an Earth-sized planet in an Earth-like orbit about a sun-like star. Right now, that is beyond our ability. We cannot find another Earth because our sun is so big, so massive, so bright compared to like what's right beside it, another Earth. So that's the one I'd like to study. We have NASA's Habitable Worlds Observatory that may be decades away. We have ideas of how to find those planets sooner using ground and then followed up by space-based telescopes. So there's that. But in the shorter term, we're trying to study what we have. And talking about going beyond Earth, you know what the easiest kind of planet to study right now? It's called a sub-Neptune, planet that's smaller than Neptune, but bigger than Earth, about twice the size of Earth, that is the most common planet we can find so far in our galaxy, but we have nothing like it in our solar system. So I'm not saying, I want to, would be my favorite planet to study. I'm saying it's what we can study now. And maybe they have hot water oceans, maybe they have water clouds and there's life in the clouds. So really, we just want to find anything right now.
Okay, appreciate that. This question is for Dr. Talley. How have lab experiments improved over the last decade?
Yeah, so I've talked about a couple of these examples already, but lab experiments have come a long way since I was in grad school. I wasn't doing molecular biology, but a lot of my friends were, and they were, in the lab pipetting things. If they were studying something on a particular time scale, they would be coming in at 2 in the morning, and then 4 in the morning, and then 6 in the morning. If the power went off, they would lose their cell culture. So I think automation, which I talked about before, has changed things substantially in being able to automate a lot of these processes to free up some of that labor. I think the 2 Nobel Prizes I mentioned have contributed substantially, though there's more work to be done. So it's AlphaFold 2, which predicts that, what is the form of the structure from the sequence. And I think there's a lot more to be done there in expanding that type of prediction capability. And then the genetic manipulation, right, has come a long way with CRISPR-Cas9, in being able to target rather than just do random mutations to phenotype, and figuring out what that phenotype was and doing all of your crosses over generations and hoping for the best. So having that targeted genetic manipulation has substantially changed things. I would love to see us move away from model organisms a little bit to things that are non-model, right? So again, back to what can we engineer versus what can we find? There's a lot of polyextremophiles where life for them is very, very different than, the E. coli or the yeast. That's sort of that lab model species that everyone cultures, everyone uses. And so I'd love to see what we call culture omics take some huge advances forward. I think that sequencing, that cost really needs to come down. It's cheaper, but it's still very expensive to actually do the sequencing. So there's a lot of what's called genetic dark matter out there. And even among species that are well known, the annotation of proteins to function is not 100% in, I think, in any species. Even in coli is only 70% annotated, where we know exactly what that sequence does in its function in the cell. And so there's a lot to be done.
Perfect. As long as we've got you up, this next question is for you as well. You discussed bioengineering to make materials in space. What considerations do we need to take into account when sending biology from Earth into a space environment? Is planetary protection a concern?
Oh, I'm so glad someone asked this question. Everyone knows the military loves its alphabet soup, its acronyms. And if you talk to almost any program manager or program officer at any of the agencies, we will have to discuss and answer questions about what we call ELSI. ELSI is E usually, depends on who you talk to, but it's the ethical, legal, societal, and perhaps environmental impact of whatever you are doing. So a lot of funding agencies, DARPA is a good example, they'll actually have an ELSI panel of members that they consult on all of their projects to talk about every single element of that acronym, because it is so important that the work that we do does not negatively impact our environment, doesn't have any legal implications. And so, yeah, I alluded to this, I think, in talking about the challenge to homeostasis, but I didn't explicitly call this out. So if I'm sending up a living engineered material, for example, that's on a satellite that can self-heal from micro debris collisions, we need to know what is that material going to do over the life of that satellite. And I think then it also speaks to the mess that we have created and that we are still contributing to in terms of the junk that is in low Earth orbit. It is a continuing, ongoing problem. So how do we address the life cycle management of anything we put up into space, living or not, and making sure that's not negatively impacting anyone here on Earth, any of the terrestrial bodies that it may come in contact with. So it's a very important concern, and it's something that we talk about and pay attention to quite a bit.
Thank you. Very, very important points. This next question I'm going to direct to Dr. Dworkin, but again, I'd like anybody else who has thoughts here to weigh in, please. NASA's Planet Hunter program brilliantly tapped into a unique human superpower for detecting patterns of light fluctuation in deep space images that suggest exoplanets. What might the next citizen scientist mission in space look like?
Well, I'm the wrong person to ask this sort of question, but I can point to something in the past, which is on the Osiris-Rex mission, they're looking for a place to get the sample from, as well as doing bolder statistics. We employ citizen scientists to count rocks and measure their length, and I also participated to generate a large model, a large database of citizen scientists who then helped us categorize the boulder statistics on Bennu, and also helped us contribute to a machine learning model to help pick out potential sampling sites. We actually used one that was picked by human eyes, but we did that as well. But there are I'm not an expert on the citizen science work. Otherwise, I used to participate in SETI at home. I don't know if that still exists. There's also a galaxy defining a thing, and I wasn't very good at it, but there was one on the Stardust mission looking for particle tracks in the aerogel on the interstellar collector. But my colleagues probably have better answers than that.
Anybody else want to weigh in on this?
I just want to say I agree with the commenter how popular it is. I was part of the NASA test mission for planet finding through transits, and the team would put the data onto planethunters.org and it was gone like so fast. People churned through that data. And the crowdsourcers were amazing. They helped find things that people had missed, actually, the professionals had missed, and it was hugely successful. I do want you to know how much effort is behind it. I personally didn't do it, but like Dr. Dworkin, it's so much work to like make it all uniform, get it ready, answer questions. So that's probably why we're not seeing as much of it as the crowdsourcers would like.
And there also are regulations on collecting data from the public and making sure that it's used ethically and etc.
Okay, Dr. Seager, this next question is for you. Before we're able to do in situ observations in the atmosphere of Venus, what observations from Earth could help increase the confidence of a positive detection of phosphine?
Not much, unfortunately. The thing is that Venus's clouds are so thick, they're like 20 kilometers deep. And if we want to look for phosphine at other wavelengths, we simply can't penetrate the clouds there. We actually did try. Several people tried looking at the near infrared, where phosphine also has it has a vibration rotational band, and you can't really. But in the radio, that's where the original detection was made. We could try again, and people are trying to make the observations again. So it's kind of, unfortunate. But in science, sometimes we just have to live with uncertainty for quite a while.
Fair enough. I'm really not sure who to throw this one at, so I'm just going to toss this one into the ether, and anybody who wants to pick it up can pick it up. Could a machine learning AI program go through all elements in the periodic table to see what chemical combinations could occur and their physical state, solid, liquid, or gas? And can those mimic Earth life chemical processes?
Like, we know that we're not there yet, right? Because AlphaFold can do one thing. It can tell us how, if you give it the monomers of amino acids, how it will fold and what it will look like. We heard from Jennifer already that it's not doing function. It's not telling us how these things are functioning. So AI is so far from being able to do that. That's not to say it's not a good idea, but there'd have to be a lot more done. That definitely is, honestly, probably beyond 2050.
Anybody else want to weigh in on this?
The periodic table is easy. There's only less than 100 elements, and only of those, only about 90 are stable enough to be considered. So that's straightforward. It's the organic chemistry. When you start making compounds, even when you start making minerals, there are 4,000 minerals and millions of organic compounds that are known. So you need, instead of having a garbage in, garbage out, you need to supply your machine learning, your AI with real data based on real experiments to be able to make these predictions. And so this gets back to what Dr. Talley was saying, that we need to do the low risk, boring experiments that no one wants to fund, but need to feed all of these models.
I mean,
It's
people do
fundamental chemistry.
for drug discovery. People do put all the elements together for drug discovery. You can make like 10 to the 200 molecules. You wouldn't want to, but you make some giant number of molecules and then sort through them in your computer. But how those all interact with each other and how they're interacting with the environment, we someday, hopefully, but not, we're very far from being able to do that right now.
Yeah, you're straying dangerously towards my expertise now on the drug discovery side. Yeah, that in silico compound discovery has really replaced the sort of prospecting for drug discovery that we used to do 25 or 30 years ago. But it is an incredibly compute intensive and laborious
Because
process.
there's so many possibilities, right? That's
Absolutely. Yeah. It's a
Yeah,
wide search space.
I think if we just went forward with this type of in silico experimentation right now and stop doing these fundamental chemistry and biology experiments to get more data in different areas, then we're leaving behind a huge amount of potential potential outcomes, right? So AlphaFold 2 is great in some cases for particular proteins of a particular size, right? But it leaves out proteins of a different size, that are much, much larger, proteins that combine and have a different action depending on the conditions. And I think Dr. Seager was speaking to that with chemistry as well. And so, yeah, leaving behind that bench level experimentation would be a huge loss to the future potential. I think the advancement, yes, needs to go towards getting that more, getting that data, but also how can we speed that up? How can we do those experiments, automated? How can we do them faster and cheaper?
Yeah. So furthermore, doing in silico experiments looking for a specific drug, which is defined by a chemical interaction that we can understand. That makes sense. The problem is we don't have a good chemical definition of life. We know life when we see it, but we can't define it chemically. All of our definitions fall short. And so we don't even know what the question is, really. And so how do you define a computer model to answer that question? And you'll get 42, but what's the real question?
But you can see why Dr. Dworkin, Dr. Talley, myself, this is the question we've got most excited about, because we want to do this. We like the question. We want to see this happen. And we're starting to see in some areas of material science how they're bringing everything together. They're making the new measurements. They're using, trying to automate and make all the measurements faster. They have to get data before they can plug into the machine learning. They're doing traditional, they're doing new, they're doing some ab initio, quantum computation. So it's all good.
Okay, we are getting close to time now for our last quick topic. I want to go through final remarks. We're just going to go again round robin for the panel. Each of you take a minute or two to identify a topic for a future science fiction novel or film that would encourage young researchers to follow in your footsteps. Not really sure. This is a free form question. Take it where you would like to take it. Dr. Bergner, why don't we start with you?
I was really hoping you would go in reverse order for this one.
Sorry, got to start somewhere.
I feel like my mind is too polluted by all the Star Trek that I've seen. But yeah, there's just, it's hard to come up with something that hasn't been done by Star Trek. But no, I mean, I think the possibilities for like some of what we've touched on today of what life would look like under sort of different environments is just intrinsically really exciting to think about.
Okay, and I'm taking notes for my next book here, so please try and make this as detailed as we possibly can. Dr. Dworkin, what about you?
Well, I guess Star Trek has never used a tricorder to check the chirality of the organisms on the planet to see that it has the right chemistry, the right compounds, but the opposite chirality. Not people get confused between left and right chirality and matter and antimatter. If you have left and right chiral molecules touching, they don't explode. They just taste differently. Some might be poison, some might be good for you. Expanding, actually, one thing that I would love to see is a good science fiction explanation of what organic is. It's very hard to explain to the public, oh, I study organic chemistry. Oh, that means expensive, not used with pesticides? No, that's organic vegetables. I mean, chemistry made with carbon. Oh, like steel? Well, not exactly. Like carbon dioxide? Well, no. Is cyanide organic? Probably. It's you can get mired in the minutia as a chemist, leaving the public behind. And so a way to explain chemical diversity, chemical reactions, what organic chemistry is, would be welcome, so I can reference it.
I'm not sure I could get my agent to go for that one, but I'll take it under consideration.
Just a subplot.
Fair enough. Dr. Seager, thoughts?
Can you just repeat the question, please?
I could repeat the question. Yeah, identify a topic for a future science fiction novel or film that would encourage young researchers to follow in your footsteps. So what's a plot for a movie that could make people want to be you?
Let's see. Well, I have got the opportunity to work on a science fiction story, so I can point you to that. It's called Etchings. But it's the idea out, this of course has to do with Venus and the atmosphere, and it has to do with bacteria also being intelligent, like a swarm of ants. So thinking about other life forms out there that are kind of more distributed.
Okay. All right. We can work with that. Dr. Talley, what about you?
Yeah, so I like the distributed sensing. I also like augmenting our own sensing. I feel like we are sometimes so very stuck in how we sense and perceive and think about the world that it's really hard for us to be creative and change that. And so I don't eat a lot of meat, and I definitely don't eat octopus anymore, ever since learning about how their nervous system, along a completely different selective, evolution pathway, have come up with an intelligence that we really still don't understand. And I think part of the reason we don't understand it is because we are too anthropomorphic. We are too stuck in thinking and seeing the world as a human. And so I think if we could augment our own sensing and understanding, that would be incredible to see and understand the world in this different way. And then bringing it back to what my career has always been about, my education, is once we understand it, how can we then use it? So what would an octopus in space, be doing and thinking?
I'm going to just really quickly recommend you take a look at the works of Adrian Tchaikovsky. He specifically has a book about octopuses in space and what they're thinking about. It's really brilliant work. So give it a look. He's a master at thinking of what other non-human minds might be and how they would work.
Interesting. So I've thought about what would it be like if sight was not our primary sense for exploring the universe. Suppose it was smell or taste. How would we design our scientific experiments and read our spectral data by taste alone? If that was
then I have a book recommendation for you, and that's is it Children of Time? I'm sure Ed can correct me here.
That's the series I was referring to. That's Adrian Tchaikovsky.
Yes.
Absolutely. Yes. Yeah. Yes, indeed. All right, that's a good place to wrap. I'm going to hand things back over to Mike now for closing remarks. Mike, you there?
Oh, sorry, I forgot to hit. I was muted. Thanks, everybody. It was a fantastic discussion. I'm glad some of my favorite topics got hit on. Protocells, chiral chemicals, and how to get samples back from space. And again, like Jennifer said, a lot of this reminds us of Star Trek and their tricorders. So thanks, everybody. Stay tuned. Sign up at our website for updates, and we'll follow on with 2 more webinars on chemistry in 2050 in the fall.